Imagine a bank robbery, right? Usually it's this loud, chaotic event. The alarms are blaring, the vault is blown wide open.
There are muddy footprints everywhere.
Yeah, exactly. Muddy footprints. It's messy,
Mm.
but it's entirely obvious what just happened. The
Mhm.
police arrive, they see the damage, track the thief, and just close the case.
Right. It's a very visible, linear chain of events. Cause, effect, and resolution.
But consider a scenario where the alarms are screaming,
Yeah.
the bank tellers are in a total panic, but the vault looks completely untouched.
Oh, right. Like an invisible crime.
Yeah, the doors are locked from the inside, there are absolutely no footprints, and the police they can't find a single trace of an intruder. The bank is slowly, systematically being dismantled from the inside out, while the police just stand around in the lobby.
Completely unable to stop it. That perfectly captures the diagnostic nightmare of the pathogen we are analyzing today.
It really does. Welcome to today's deep dive, everyone. We are digging into a massive stack of clinical guidelines, microbiological research, and immunological studies focused on an emerging sexually transmitted bacterium called Mycoplasma genitalium.
Often shortened to MGen.
Right, MGen. And the mission for this deep dive is specifically tailored for you. We are going to unpack exactly how a persistent, low-grade infection with this microscopic stealth pathogen alters the mucosal inflammation pathways in the female reproductive tract.
And critically, how it triggers real severe physical symptoms.
Yes, specifically deep dyspareunia, which is intensely painful intercourse. So to understand the severe physical pain this bacteria causes, we really have to understand how it operates with almost no tools.
Which is fascinating. When we talk about MGen, we are talking about an organism that boasts the smallest genome of any known self-replicating bacterium.
Because saying it has the smallest genome might not sound that intimidating, right? Not until you look at the actual numbers.
Oh, the numbers are wild. We are talking about roughly 500 genes.
Only 500.
Yeah. To put that in perspective, a standard bacterium like E. coli, which most people are familiar with, has over 4,000 genes. Human cells have upwards of 20,000.
Wow.
So MGen is the ultimate biological minimalist. It operates with a genetic toolkit so incredibly limited that, biologically speaking, it shouldn't even be able to survive, let alone outsmart the human immune system for years.
It's discarded almost everything throughout its evolution, right?
Exactly. Most notably, it completely lacks a cell wall.
And a cell wall is usually the defining armor of a bacterium. It gives them their shape. It protects them from the environment.
Right, and because it lacks that armor, and because of this extreme genetic reduction, MGen cannot synthesize its own amino acids. It can't build its own fatty acids.
It can't make anything it needs.
Basically, no.
Yeah.
It can't even synthesize nucleic acids from scratch to replicate its own DNA. It is entirely 100% dependent on the host environment for its basic survival nutrients.
So it's an obligate parasite. It has to steal from us just to exist. It's like a master thief who breaks into a house bringing absolutely no tools. No lockpicks, no blowtorch, no rations. They rely entirely on what they can scavenge from the homeowner's own kitchen to survive.
I like that. And taking that master thief analogy a step further, they also need a way to hold their ground once they get inside.
Right. They can't just float around.
No, because
Yeah.
the female reproductive tract is lined with mucosal fluids designed to wash away debris and invaders. If MGen doesn't hold on tight, it simply gets flushed out.
So how does it hold on?
To solve this, it uses a highly specialized structure called a terminal tip organelle.
I'm picturing like a microscopic grappling hook.
It acts very much like one, but the chemical mechanism of this hook is just a brilliant piece of biological hacking.
How so?
Well, MGen takes an enzyme that normally functions inside its own cytoplasm, specifically one involved in basic sugar metabolism called GAPDH.
Okay, an internal metabolic enzyme.
Right, and it somehow manages to push this enzyme out onto its surface.
Wait, really? It's using a metabolic engine part as an exterior tool.
Yes. It's called a moonlighting protein in the research. Once on the surface, GAPDH moonlights as a molecular glue.
Oh, wow.
It binds incredibly tightly to human vaginal and cervical mucin, which is that protective gel-like substance lining the reproductive tract.
Okay, so it's stuck to the mucin.
Yep, and it also binds to fibronectin, which is a major structural protein that helps hold our tissue cells together.
So it's literally super gluing itself to the walls of the house using a repurposed sugar enzyme. That is incredibly resourceful.
Resourcefulness born of absolute necessity, right? Because by using GAPDH as an external adhesive, MGen establishes this firm, immovable foothold on the mucosal epithelial cells of the lower reproductive tract.
Okay, so our thief has broken in, scavenged for survival and glued themselves to the wall.
Huh.
But looking at the research, MGen grows very, very slowly, doesn't it?
Glacially slow.
Yeah.
A colony takes weeks to form in the lab, which is why it's so incredibly difficult for doctors to culture and diagnose.
Right. So it's growing at a glacial pace. How does it cause such immense pelvic pain? I mean, the destruction isn't primarily caused by MGen directly eating away at the tissue, is it?
No, the twist here is that the tissue damage isn't caused by MGen directly. The destruction is overwhelmingly caused by our own immune response.
Like a massive case of friendly fire.
Exactly. It's a prolonged, severe case of friendly fire.
So walk us through that. How does this silent, slow-growing, tiny bacterium trigger a destructive immune response that basically ruins the host's tissue?
It all goes back to the fact that MGen lacks a cell wall.
Right, no armor.
Yeah, in a normal bacterium, that thick cell wall acts as a barrier, shielding the delicate internal membrane from the outside world. But MGen's cellular membrane is completely naked, exposed directly to the outside environment.
Okay, and why does that matter?
Because this membrane is heavily loaded with specific molecules called lipid-associated membrane proteins, or LMPs.
LMPs, so these are essentially the chemical signature of the bacteria.
Precisely. When these exposed LMPs come into physical contact with the host's mucosal epithelial cells, and remember, MGen is anchored tightly right against them using that GAPDH glue, they are immediately recognized by the host's surveillance system.
Our cells have sensors.
Yes, our cells are covered in pattern recognition receptors. They are like outer perimeter guards looking for anything that doesn't belong. Specifically, MGen's LAMMPs bind to toll-like receptors 1, 2, and 6 on our cells.
Toll-like receptors, or TLRs. So these are the sensors waiting to detect an intruder's chemical footprint.
Right. And when TLR 1, 2, and 6 are triggered by the bacterial AMPs, they send this frantic chemical signal deep into the nucleus of the human host cell.
Okay, sounding the alarm.
It flips a master genetic switch known as NFKB.
I saw NFKB all over the immunology studies. What happens when that switch is flipped?
NFKB is a transcription factor. You can think of it as the ultimate panic button for inflammation. Once it's activated, the host cell starts reading emergency genes and pumping out massive amounts of pro-inflammatory cytokines.
The distress signals.
Yes, in the research, you'll see a specific cast of characters: interleukin-1, interleukin-6, interleukin-8, and tumor necrosis factor alpha.
So these cytokines are like chemical flares shot into the tissue.
They are distress signals that drastically change the local environment. They cause blood vessels to dilate and become leaky.
Which brings heat and swelling to the area.
Exactly. And the cytokines also act as a homing beacon, calling in a swarm of white blood cells, particularly macrophages.
Macrophages being the immune system's heavy infantry designed to swallow up and destroy bacteria.
Yes, this is the textbook definition of acute inflammation.
Okay, wait, hold on. This is where I have to push back because it seems like a logical paradox.
Go for it.
If the mucosal cells are successfully sounding the alarm, the NFKB switches on, the blood vessels are dilating, and the cytokines are actively guiding macrophages into the area, shouldn't the immune system just sweep in, eat the bacteria, and end the infection?
It seems like it should, right?
Yeah. I mean, if the alarm is ringing that loudly, why doesn't it clear the bacteria? Why does this become a chronic issue that plagues a patient for months?
That is the exact paradox that makes MGen so insidious. The alarm is indeed ringing. The cytokines are flooding the area. The macrophages have arrived, but the intruder has found the panic room.
The panic room being, wait, inside the human cell itself?
Exactly. MGen doesn't just sit on the outside of the cell waiting to be eaten by a macrophage. It actively forces the host cell's internal scaffolding, the actin cytoskeleton, to rearrange itself.
Oh, wow.
It tricks the human epithelial cell into reaching up and swallowing the bacteria whole, pulling it inside. This process is called endocytosis.
So it basically forces the host cell to eat it, but not to destroy it.
Right. MGen sets up a permanent camp inside the host cell. It can survive inside membrane-bound transport vesicles. It can bring out and float freely in the cell's cytoplasm.
Just living inside our cells.
And astonishingly, researchers have even documented MGen localizing inside the host cell's nucleus, hiding right among human DNA.
That is terrifying. So by physically hiding inside the very cells that are sounding the alarm, it becomes invisible to the arriving immune forces.
Yes. By internalizing itself, MGen is completely shielded from circulating antibodies floating in the bloodstream. It's hidden from the complement system, which is that network of blood proteins that punch holes in pathogens.
Right.
And most importantly, it's safe from the macrophage cells that are swarming the tissue because of the cytokine flares. The immune police are in the building, heavily armed, but they can't find the thief who is hiding inside the walls.
But the host cell isn't completely defenseless once the bacteria is inside, though, right? Doesn't the human cell try to kill what's inside it using its own internal defense mechanisms like oxidative stress?
It certainly tries. The host cell launches oxidative attacks, essentially releasing reactive oxygen species.
Which is like internal chemical warfare aimed at damaging the bacteria's proteins and just ripping it apart.
Exactly. But MGen has this extraordinary evolutionary countermeasure. Once it is hidden inside the host, MGen releases an enzyme called MRA, which stands for methionine sulfoxide reductase.
MRA, that's an antioxidant repair enzyme, right?
It is. It literally repairs the MGen proteins that have been damaged by the host's oxidative attack.
You're kidding.
No, it restores their biological activity in real time. The host cell throws a chemical grenade, and MGen just patches up the shrapnel damage and keeps functioning.
So the immune system is essentially bombing its own city because it can't clear out the sniper.
That's a great way to put it. And to make matters worse, MGen actively manipulates the host cell's apoptosis pathways.
Apoptosis being programmed cell death.
Right. Usually when a human cell realizes it is infected and it just cannot kill the invader, it will sacrifice itself. It will undergo an orderly death to destroy the pathogen inside and prevent the infection from spreading.
A scorched earth defense protocol.
Exactly. But MGen secretes factors that interfere with this death signaling. It forces the infected host cell to stay alive against its will, keeping it functioning just long enough to serve as a continuous, safe shelter and nutrient source.
Let me make sure I have this straight. The host cell detects the bacteria via toll-like receptors and pumps out inflammatory cytokines. But the bacteria hides inside the cell, repairs any oxidative damage using MSRA, and then literally stops the cell from committing suicide.
That is exactly what happens.
The result is this incredibly frustrating biological loop. The immune system is constantly triggered, the alarm is always ringing, but it cannot eliminate the target.
The inflammatory switch is stuck in the on position indefinitely. It creates a state of chronic, unresolved mucosal inflammation. The host tissue is in a permanent state of emergency.
So if you're looking at a patient's chart, or if you are someone experiencing this, the core question is how this microscopic intracellular hiding game translates to severe, debilitating physical pain.
Right.
How do we get from a stuck NFKB switch in a confused macrophage to deep dyspareunia?
To understand the pain, we really have to track the bacteria's movement anatomically. MGen rarely stays confined to the lower reproductive tracts like the vagina and the lower cervix.
It moves.
Yeah, over time, because it is not being cleared by the immune system, it travels upward. It migrates into the upper cervix, into the endometrium that lines the uterus, and critically, up into the fallopian tubes.
So it crawls all the way up into the upper reproductive tract.
And as it migrates, it brings that chronic inflammatory loop with it. Remember those cytokines we discussed earlier?
The chemical flares. IL-1, IL-6.
Exactly. The constant, unabated presence of those chemicals, particularly interleukin-6 or IL-6, begins to severely shift the local tissue environment. IL-6 is known in immunology for transitioning tissues from a state of acute, short-term inflammation to chronic tissue remodeling inflammation.
Wait, tissue remodeling. So the tissue itself actually starts to change its structure because the immune system won't turn off.
Yes, and the damage is most pronounced and devastating in the fallopian tubes. The fallopian tubes are these delicate, narrow passages lined with cilia.
Cilia being the tiny microscopic hair-like structures that beat in a rhythmic wave to help move the egg from the ovary down into the uterus, right?
That's them. When MGen reaches the fallopian tubes, it specifically targets and attaches to these ciliated cells. The toxic immune response, the constant bombardment of inflammatory chemicals, causes those delicate ciliated cells to abnormally swell.
Ouch.
And eventually, they shed off the lining of the tube entirely.
Destroying the very mechanism meant to transport the egg.
Yes. And without the cilia, fluid begins to pool. This leads to a condition called tubal edema, where the fallopian tubes become swollen, blocked, and heavy with fluid.
Like a backed up pipe.
Very much so. In clinical terms, a fluid-filled, blocked fallopian tube is often referred to as a hydrosalpinx. The tissue is quite literally being degraded, stretched, and severely swollen by chronic inflammation.
This upward spread of the bacteria, combined with the resulting tissue destruction, swelling, and fluid accumulation, this is the core of what clinicians diagnose as pelvic inflammatory disease, or PID.
Exactly. PID isn't just a single symptom, it's a broad clinical syndrome of this upper tract inflammation and all the resulting anatomical chaos.
Okay, so we can finally connect the dots to the physical pain, the clinical manifestations of this chronic, low-grade pelvic inflammation, the literal physical swelling of the fallopian tubes, the shedding of the uterine lining, the constant heavy cytokine wash. This is what turns the pelvis into a hypersensitive environment.
It really does. The uterus, the cervix, and the fallopian tubes are all tethered deep within the pelvic bowl. They aren't just floating in space, they are anchored by ligaments.
Right, because all of these organs are anchored deep inside the pelvis. Any physical impact or upward pressure is going to be felt intensely.
Right, and during intercourse, there is mechanical upward pressure on the cervix and the lower uterus.
So intercourse physically pushes on an environment that is already stretched to its absolute limit.
Yes.
The microscopic tug-of-war has turned the entire pelvic region into a bruised, fluid-heavy, swollen landscape. The thrusting or pressure acts directly on organs that are suffering from tubal edema and continuous pro-inflammatory cytokine bombardment.
Exactly. It is not a functional pain, and it is not a referred pain where you feel it somewhere else. It is direct mechanical collision with tissues that are heavily inflamed, bloated with fluid, and suffering from active cell death.
That sounds excruciating.
It is. The nerves in those tissues are highly sensitized by the continuous presence of cytokines like IL-6. Every movement registers as acute physical trauma. That is the explicit physical mechanism behind severe, deep dyspareunia.
It is an absolute masterclass in microbial manipulation. The destruction isn't caused by the pathogen's sheer force, but by its ability to turn our own defenses against us.
So true.
Let's trace the full journey of what we just explored today. We started with a minimalist bacterium equipped with only 500 genes. It uses a repurposed internal metabolic enzyme, GAPDH, to superglue itself to mucosal cells and fibronectin.
And because it has no protective cell wall, its exposed lipid proteins, the LAMPs, hijack the host's toll-like receptors on the cell surface, unleashing a massive cytokine storm and triggering acute inflammation.
But then it evades the very storm it created. It tricks the host cell into swallowing it through endocytosis.
Right.
It repairs any oxidative damage thrown at it by using the SRA enzyme, and it secretes factors to stop the human cell from dying, basically settling in for a long, hidden siege.
And that long haul allows the bacteria to slowly migrate upward into the uterus and fallopian tubes, dragging that chronic IL-6 inflammation with it.
Where it destroys the delicate cilia, swells the fallopian tubes with fluid, causes pelvic inflammatory disease, and transforms the pelvic cavity into a bruised, swollen environment, making the mechanical pressure of intercourse intensely, deeply painful.
A macroscopic physical nightmare entirely driven by a microscopic ghost hiding in the walls.
Before we wrap up this deep dive into MGen, there is one final chilling thread hidden deep in the research material that we want to leave you with to ponder. It goes beyond the immediate pain and addresses the long-term shadow of this infection.
It's a very important thread because it connects back to the fundamental nature of chronic inflammation. If we look at the bigger picture of human biology and oncology, we know that long-term, unresolved chronic inflammation and cellular immune evasion are two of the primary drivers of cellular transformation.
When tissues are constantly bathed in toxic inflammatory cytokines for months or years, when they are constantly trying to repair themselves, and when they're constantly being forced to stay alive by bacterial signals preventing normal apoptosis, the cellular machinery just gets tired, right?
Yes. Mistakes in DNA replication happen. Things can go wrong.
Extremely wrong. Given that MGen causes this highly prolonged low-grade inflammatory state, researchers started looking at its impact over decades. It has actually been noted in some recent studies to alter stem cell properties and induce malignant transformations of cells in laboratory settings.
Which makes sense biologically. If a bacterium can disable a cell's ability to undergo programmed death while simultaneously forcing it to live in a highly oxidative, inflammatory environment, you are looking at the exact recipe for tumor growth.
So what is the true long-term link between a persistent, undiagnosed MGen infection and the development of reproductive tumors, like ovarian or cervical cancer, years or even decades down the line?
It's a sobering question, and one that researchers are only just beginning to unravel. It completely changes how we view asymptomatic or slow-growing infections. A low-grade infection doesn't mean a low-grade threat.
It takes us right back to that bank robbery analogy. The thief didn't just break in, set off the alarms, and hide in the walls. They moved in. They changed the locks to the panic room. And over the years, they might just be quietly remodeling the entire structural foundation of the house without anyone noticing until the walls inevitably cave in.